Let's begin today with what I think is the most fun part of the class, which are the way in which microorganisms interact. But what we're going to be looking is pathogenesis, which is, as you know, the influence of disease. So what I'm going to begin to bring up to you, there we go, well, it's that every time that we come in contact with a microorganism and they either are growing in us or growing on us, that is considered an infection. And again, an infection doesn't have to be detrimental. Those of you, we discussed the issue that the plant gets infected by the rhizobium to establish a beneficial relationship for nitrogen fixation. So everything has to do with the exposure to the organisms is infection. So as we know from that example, most of the relationships that we have with the microorganisms tend to be beneficial. The microorganisms that we have in our gut, the organisms that the plant has, the ones that we discussed about the symbiotic relationship, which are positive, like the mycorrhizobia with the plant, roots with the fungus. So the problem comes when there is actually damage to the host, as in the case of parasitism, because that is what we consider disease. Disease, it's the host microbe interaction that is going to result in some kind of damage from parasitism. And what we're going to learn is that there are going to be multiple facets to disease. So we tend to call them pathogens. Every microorganism that causes disease is a pathogen. They can also be called parasites because they are now going to be in a parasitic relationship. So remember, symbiotic relationship could be beneficial but could also be harmful. And in this case, parasitic relationships are causing some damage to the host. Now, when we think about those relationships between humans and microbes, we're going to think about it in multiple terms. We're going to look at primary pathogens. We're going to look at opportunistic pathogens. And in the case of primary pathogens, these are going to be a microorganism that is going to cause some level of pathology within the host. We are going to be quote-unquote normal hosts, all of us. And again, when we think about pathology, that is any condition that deviates from normal. And that is why it's defined based on quote-unquote the person's normality. And it is going to be very specific for disease. Now, an opportunistic pathogen, it is a pathogen that is able to induce a level of pathology when the host immune response is not working appropriately. So this came out to be very important, for example, in the case of HIV-infected individuals that were usually not being harmed by HIV, quote unquote. They were dying out of opportunistic infection that they have with other pathogens that the immune response will normally keep at bay. Pneumocystis carinii, hepatitis virus 8, which is the one that causes sarcoma. So any patient that for some reason his immune system is now compromised, either because that person has HIV or that person is undergoing chemotherapy for cancer or that person is in any other way, for example, for a transplantation, the person will get drugs that are going to attenuate the immune response so the organs will not be rejected. All of them will succumb to pathogenic opportunistic infections. Now, when we think about how aggressive it's a pathogen, we think of the term as virulence. So it is a quantitative measure of how aggressive a microbe is. And we then do experiments in animals to determine the number of microorganisms that are needed to give you disease in a particular time. And what we're going to see is that the amount of virulence is going to mediate that relationship between the host and the pathogen. For example, we can take a very virulent pathogen and attenuate that pathogen, meaning that we take away the virulence and use that pathogen now as a vaccine agent. So we have diminished the virulence of the pathogen, but are now able to use it because it's still alive and use it as in a vaccination with a live vaccine. So now, when we think about how that relationship is going to, the outcome of that relationship is going to be between us as the host and the myriad of pathogens that are present outside, we have to take two things into consideration. One of them is going to be how virulent the pathogen is. And the other thing is how resistant the host is to that parasite. And the resistance is going to be measured by the immune system of the host. All of us being genetically different, we have different resistances to microorganisms. So now, why are we germ magnets? Why do germs love to live on us and in us? Well, we are a nutrient-rich environment. We're constantly sweating. I mean, microorganisms that are in our outside, they're dead cells that could be degraded down. We have sweat. We have other things that we are secreting that could be like the sebaceous secretions that could be used as source of nutrient for a microorganism. We have a constant pH, and of course we have a relatively constant temperature. Our core is more constant than our limbs, but still, we still have a constant temperature. So we have many environments that are beneficial for a microorganism to live. We're going to see that most of the organisms in our skin are going to be gram-positive microorganisms. About 75% of them are gram-pos. And the skin usually is relatively dry, but if you think about the area of the crotch, the area of the underarms, those are now wet, more moist areas that some microorganisms will find them more suitable for the life lungs. Certain organisms like to live in our oxygen-rich environments like our lungs. So for example, Microbium tuberculosis. And other ones are going to live in now completely opposite environments that are very anaerobic, like our gut. And that will be a great place for Clostridium organisms to live. So now, this image over here, what it's basically showing you is all the myriad kinds of microorganisms that we can find all over our bodies. And we have microorganisms that are going to be mostly exposed to areas that are exposed to the environment. So everywhere that we respire, so mouth, nose, sinuses, trachea, lung, that's going to be an excellent environment for microorganisms. Or digestive tract, we're constantly putting things on our mouth. We put our pencils, besides eating, we put food, that's eating. Besides eating, we put pencils, we put straws, we put our cell phones close to our mouth, all that can bring microorganisms to those areas. Of course, we have an entire skin system that is separating us, our eyes, our membranes, and then we have the system that in general we call the mucosa. The mucosa is going to include everywhere that we have a single or multiple layer of mucus. So we have the nose, the mouth, the urogenital tract, the intestine. Now, this figure over here shows you very different types of microorganisms. Again, I'm not putting this for you to memorize what organisms are where. No, you can always look into this. But what I'm basically showing to you is that you have very different species of microorganisms. We are not colonized by only one species. And another thing that I want you to bring into mind is that a lot of this depends also on the exposure that we have. We're going to be looking that for an infection you have to have exposure. So me living in the Caribbean, I'm going to be exposed to different microorganisms than me living in Alaska, or living in sub-Saharan Africa, or living in somewhere in Asia. So you have to think about the place where the organisms are present to catalog then where those organisms are going to be allow you to get. So you gotta have different levels of exposure. So here it's part of the mucosal area. As I mentioned to you, this is an epithelial area where you have some level of mucus. So again, think about the entire respiratory tract system, nose, sinuses, trachea, lungs, the entire digestive system, mouth, esophagus, stomach, large and small intestine, as well as the urogenital tract. It's going to be an area of mucosa. Those of you who will take immunology in the future, there is a specific mucosal immunity branch of immune research because these areas are constantly being exposed to organisms, so we have a really intense defense mechanism to sample what's out there and to also protect us from the environment that we're living in. So now when we think about epithelium, The epithelium, as you can appreciate from this image, it's a tightly packed layer of cells. Depending on the type of epithelium, it could be as simple as a single layer of cells, like the one in the intestine. There's going to be a lower area here called the basement membrane, and those cells basically, it's the only thing separating the environment from the body. So think about the small intestine with a simple epithelia. The mucus is designed to give an area of protection, and it's basically a layer of soluble glycoproteins produced, in the case of the intestine, by M cells, M because they're mucus cells, or goblet cells, excuse me. The goblet cell is the one producing that. And the goblet cell produces this mucus, and the mucus has compounds that are going to be antibacterial. It's also areas that are constantly in flux. Think about all the ways that we swallow. We're constantly swallowing and allowing movement of things. There is really not a lot of stationary environments in the mucosa. They tend to be constantly moving. We're constantly getting phlegm from the lungs that we swallow back down. We constantly get saliva that is irrigating the area of the mouth, and we swallow that back. And of course, our intestinal tract is constantly moving material. So the areas of the mucosa are going to be in constant movement to eliminate the mucus as part of the natural biology that they have. In the urogenital tract, we have a constant flux of lubricating mucus, as well as urine that comes out and helps also be a barrier. So you have to then have microorganisms that are going to have to fight those physical barriers. So the mucosa per se, it's going to be our first line of defense against microorganisms that we're going to be exposed to. And you can have microorganisms be in loose association with the mucosa. Other ones are going to be able to adhere better, so they will have to have adhesion molecules in their surface, maybe make a biofilm that is going to keep them trapped. And after that point, what we're going to see is that they can form colonies which can either invade and infiltrate or go inside a cell if they are intracellular pathogens. So now this figure is a little bit different from figure 23.1 that is on the book, because on the book it's just showing them as colonies on the top. But I decided to keep this one from a previous version of it because it's actually closer, in my opinion, to what is actually happening. So when we look at the microorganisms that are present, we can think of multiple microbial environments: skin, the mouth, the respiratory tract, gastrointestinal tract, and the urogenital tract. And as you can appreciate from this list of microorganisms, they're going to be very different from one another. We're going to see that about 75% of the skin bacteria are going to be either firmicutes or actinobacteria, so gram-positive microorganisms. Few are actually gram-negative, about 25%, and most of them are protobacteria. In the mouth, we're going to have multiple different kinds of microorganisms, but they're not only bacteria. We have a lot of yeast living in our tongue, for example. And one of the things, if we have time, we're going to be looking at some of the environments in the mouth. I know that some of you want to go to dental school, so this is going to be interesting for you. In the respiratory tract, again, organisms that are able to endure the high oxidative stress of the lungs. And in the gastrointestinal and urinary tract, organisms that are able to then have ways of maintaining their colonies in an area of high flux. Some of the microorganisms in the intestinal tract that you should be very reminded of are the enterobacteria. Entero because it's an enteric tract. So gram-negative microorganisms, E. coli, Salmonella, Shigella, Klebsiella, all those are part of the enteric class of microorganisms. And as well, the problematic ones, Clostridiums. Clostridium microorganisms, in particular Clostridium difficile, is one of the most horrible pathogens that we have that cause inflammatory bowel syndrome. In the urogenital tract, that's going to be interesting because you have differences between the male urogenital tract and the female urogenital tract, and you have microorganisms that are going to cause disease. Escherichia some of the E. coli family can infect the urogenital tract, as well as, of course, as microorganisms of the Neisseria family. But what we're going to be seeing is that you also have, all our mucosa have commensal organisms that are part of the flora that helps us maintain the biology of the mucosa. So let's look at some of the barriers of infection that are being challenged by these microorganisms that we have. And again, we can think, for example, at the area of the mouth and the nose. We have areas that are going to have cilia, and the cilia are going to be beating the mucosa to move it out. We're constantly, as we're sleeping, having mucosa from our mouth and your throat moving forward. So the cilia is going to help move that around. As you know, the skin is a physical barrier that protects the entire body from desiccation, but it also is a barrier to microorganisms. We have sweat, and the sweat has antibacterial peptides. Some of them are called defensins, and the defensins are good to fight certain microorganisms and render them harmless. And we have normal bacteria living in our skin. No matter how much you try to clean your hand with sanitizer, you do not get rid of them. They are part of our normal flora. So they are good to have in our skin because they prevent other organisms from taking hold. So having a healthy microorganism flora in our skin prevent pathogens to come in and establish infections because they are competing for the same ecological niches. So you want to have those bacteria present. Now, as we ingest food, the first place that food is going to go after the mouth is going to be the stomach. So we're going from an area of relatively neutral pH to a very acidic area. So the microorganisms need to be able to fight that acid in order to survive and colonize the more neutral duodenum. It's about pH 4 and pH 5 after the stomach, and then it gets to neutral pH in the large intestine. And as we discussed before, you have that normal flora in our gut, in our skin, in the urogenital tract that helps prevent other organisms from taking hold. We definitely need those microorganisms for our well-being. For example, in abiotic mice, these are mice which are grown in super sterile environments. When we put them there, we harvest the babies by C-sections from their mothers, and put them with mothers which are very sterile, so they do not get exposed through the maternal route to bacteria. So we're growing this mice, for example, in this super germ-free environments. They do not have a flora in their gut because they are using all the bedding, all the food has been autoclaved to kill absolutely everything. When you look at their immune system is shut. The microorganisms that are present in the gut help their immune system develop. So this idea of trying to minimize the amount of microorganisms that children, for example, are exposed increases the susceptibility for allergens and other mechanisms. So those of you who were constantly being monitored by your parents, and it's like, no, don't touch that because it has bacteria on it, that is a little bit bad for you. The kids who ate the dirt do better immunologically speaking. So that normal flora in the mice, the intestine doesn't develop appropriately. And once you colonize them with bacteria from a donor, all of a sudden the architecture of the intestine changes. Now their immune system boots up and they have better capacity of fighting opportunistic infections than the ones without the microorganisms. As I mentioned, in the urogenic tract, we have constant flushing and irrigation by urine. And that is going to help prevent infection as the liquid prevents the adherence of microorganisms from getting there. So usually the pathogens that are going to adhere to the urogenital tract have mechanisms to be able to stick on the wall of the epithelium and therefore lead to the infection. As we have talked before, the enzyme lysozyme, which is present in sweat, in our tears, there is an enzyme that attacks directly the glycosidic bond in the peptidoglycan layer of bacteria. So therefore, it helps attack the cell wall and prevents microorganisms from growing. Here we go back to the issue about the mucus. and the cilia. Again, this is now in the trachea, and that helps move microorganisms that may be embedded to it, so then we can swallow them back and digest them away. And that same thing happens now, mucous and antibacterial peptides and phagocytes in the lung. Okay, so in the lung is a very interesting place. There is a group of alveolar macrophages in the lung, and those alveolar macrophages are constantly surveilling the area and destroying bacteria that get present. They are the ones who get infected by Mycobacterium tuberculosis, because inside the alveolar macrophages, the bacteria is protected from the oxygen radicals created in the lung. That's an interesting thing. As we know, we have an immune system that is out there fighting in our behalf, both blood and lymph. You know what the lymph is, guys? Who doesn't know what lymph is? Raise your hand. Okay, so everybody knows what the lymph is. So both the blood and lymph, liquids that are having two different circulatory systems in our body, contain proteins, and those proteins are going to have antimicrobial behavior. In the blood, that protein system is called complement. And the complement is an ancient system of immune defense that is able to create pores on the surface of pathogens and basically just blow holes in them, and everything on the cytoplasm of the bacteria comes out. And of course, you have antibodies as well in the blood as well and in the lymph. As I mentioned to you, you go from a pH of 2 to a pH of 4 as you're moving from the gut to the ileum and the jejunum over here. And that also, it's a challenge for the microorganisms. If they have an adaptation to live at pH 2, all of a sudden now they have gone 2 logs change in pH, and that prevents them from and that inhibits their growth. And last but not least, again, all the epithelial layers, skin and mucosa, they have tight junctions, and because of the tight junctions, as you remember from Bio 110, microorganisms and things cannot really get in between them. So it is a really nice environment for, our body is designed basically to protect ourselves by multiple weaponry to prevent the invasion of microorganisms into our system. But we are constantly being bombarded by microorganisms, and some of them we can have a nice cohabitation with them, and other ones are actually going to be successful pathogens. So to make a successful pathogen, a microorganism has to overcome a lot of the barriers that we just saw. So they have to have access to the host and then have access to the appropriate tissue. It's not only sufficient to be exposed. The microorganism needs to have access. That's why wounds in our skin are such a easy way for organisms to then get access to the inside of us. Now, as I mentioned, we have those proteins in blood, complement as well as antibodies, which are going to be fighting incoming microorganisms. But we also have those macrophages, like the alveolar macrophages. Every tissue has macrophages, by the way. Cooper cells in the liver, we have alveolar macrophages in the lung, and the name of the ones in the skin is evading me right now. I'll get to it back to you guys later. But if a microorganism can prevent the immune system from seeing it, it can now go in quote-unquote stealth mode and therefore colonize and be able to infect first and then colonize a tissue. And that can be done by multiple ways. We're going to see some of them in the rest of the class. Now, once they are able to infect and colonize, they have to establish a replication permissive niche, meaning that they have to make a small colony and use that environment in order for them to divide. In the case of Mycobacterium tuberculosis, that environment is inside the alveolar macrophage. Other microorganisms can do it in a sebaceous gland, creating a pimple, for example. And when they're replicating, they need to be able to acquire nutrients. If they are not able to acquire the energy that they need to be able to divide, they won't be able to do so. Last, depending on the microorganism, they have to keep the host alive long enough to ensure dissemination and transmission to a new host. And that is what happens with an organism that, as I call them, have evolutionary adaptation to live in the host. For example, herpes simplex 1, it is the herpes that infects the lips, and people will get a cold sore over there. It doesn't kill us. It's able to establish a replication permissive niche in the neurons around the lip area. And because it doesn't kill us, it lets us live enough for us to kiss somebody else and pass it to another unsuspected host. Ebola, on the other hand, though is able to establish a replication permissive host, it damages the host so fast that the amount of potential hosts being exposed to it decreases as the person usually succumbs to infection really quickly. Because the adaptation of that particular virus to the host is not the same as the adaptation of herpes virus to us as host. So that is an interesting point of what makes a successful pathogen. And we're going to see that all the steps, in order for all this to happen, it's governed by virulence factors. And those virulence factors, in the case of bacteria, we're going to see that most of them are either encoded on a plasmid or on a phage that are going to be coming to the microorganism. So we're going to be looking at that for a moment. So now, let's take a moment to look at the steps that guarantee an infection. And what I want to bring up to you is that the result of the infection is irrelevant of the steps. These are the steps that are required for a rhizobium to infect a plant cell, and that is a beneficial symbiotic relationship. The same thing happens. But the same steps are going to be now used for a pathogen to infect a host that actually can cause disease. And the first step is going to be exposure. You need to have exposure to the microorganism in order for that microorganism to be able to infect you. And once you have exposure, that microorganism has to have a way to adhere to the tissue. So first thing, have exposure. After that, you have to have adhesion. Post-adhesion, that microorganism needs to invade. And the invasion can happen through the epithelium, either inside the cell or in between the cells of the epithelium cell. And after that infection, you have to form a colony and grow, and there you can also produce virulence factors that are going to help you with the steps that we have described. Now, this will happen over and over as a cycle. And these initial 4 steps are the ones which I mean that are shared among all bacteria that infect us. Either for a beneficial infection like the one in our gut, or as a pathogenic infection in the case, for example, of cholera infecting our gut. All of them are going to be the same. Now, the outcome of it, it's what is going to be different. And it's going to be this lower part over here. Because of those virulence factors that the microorganisms can have, they can have a level of toxicity. For example, a virulence factor may be a toxin. We talk about the fact that gram-negative bacteria have the LPS on their surface, and that is called the endotoxin. And that can cause fever and cause symptoms. But the microorganism could be actively producing an exotoxin, we're going to learn about them more today, in which that protein has an effect on the tissue and that can destroy it, like the necrotic toxin in the flesh-eating Staphylococcus aureus, or the ABC toxin in cholera that causes diarrhea. Now, the virulence factors are always also going to help the microorganism with invasiveness. And that is going to be, once that organism is in the gut, let's say, from the gut, it needs to move now to the blood, to a secondary tissue. For example, infections of syphilis begin in the urogenital tract of infected people, but depending on the stage of syphilis, it goes to your brain. And third level syphilis, which is the brain infection, is lethal. So the microorganism has to move from the original area of infection in the urogenital tract through the circulation, avoid every of those factors that the immune system is throwing at them, to cross the blood-brain barrier and get into the brain. So that is going to be what is going. those factors, those virulent factors, are going to mediate the invasiveness of that microorganism to allow it to go to a secondary tissue. And then in that secondary tissue, you have further damage, you have further problems, and those are the ones that cause disease as we know it. So now, let's take a look at some of these factors independently from one another. And let's begin as adherence. And we talked about this before in class. So these two slides are going to be a little bit of review. Because, as you know, microorganisms have fimbriae, they have the pila. They can create glycocalyx that allow them to attach themselves to surfaces. So those molecules present in the microorganism, in particular bacteria, are going to allow them to adhere. But there are also other proteins that are specifically made by pathogens to allow them to bind to a particular tissue. For example, Neisseria gonorrhea, it's going to create a set of proteins called OPA. And those OPA proteins are able to bind to the CD66 antigen. That is the CEA, or the carcinoembryonic antigen, that is present in the urinary tract. So that is a specific protein that allows Neisseria gonorrhea to bind to a specific protein being expressed in its niche. So it's not just a fimbriae protein that allows for general adhesion. That molecule is specific for adhesion at a specific tissue. And that is one of the virulence factors that Neisseria gonorrhea has in order to make it an effective pathogen infecting the urogenital tract. And as I mentioned, the fimbriae, the pilae, and the capsule of glycocalyx are going to be other issues that are going to benefit that adhesion. And this is what we were talking before. So remember, the fimbriae are those small protrusions that are emanating from the microorganism. They're structurally related to the flagellum, as you remember from class, but they help the organism bind in general to a surface. The pilus that we already talked about for conjugation and motility could also be used for adhesion. And then that capsule layer, it's that slime layer that is produced by microorganisms, and that serves as a glue to allow those organisms to attach themselves to surfaces. A benefit of the glycocalyx is also that it's antiphagocytic, meaning that macrophages or neutrophils that usually can gobble those bacteria without it are going to, quote-unquote, choke on them and not swallow them at all. So therefore, they are now being prevented from being phagocytosed by immune cells. So this table over here from the previous book shows you a lot of different kinds of adherence factors that are used by pathogens depending on the tissue. And in particular, remember the capsule we talked about. Here you have the villi. Here is an intestinal cell, and all those little finger-like structures are the microvilli of the intestinal cells. Notice the E. coli on it, and that E. coli has a capsule around it. So in order for that E. coli not to be flushed away with the movement of food and liquid that is present in the stomach, they are attaching themselves through a capsule that promotes adhesion to the intestinal macrovilli. Streptococcus pyogenes, which causes scarlet fever, has a protein called the M protein that allows them to bind to the respiratory mucosa. Neisseria gonorrhea, as I mentioned, has the OPA proteins. And other molecules, like in the gram-positive microorganisms, that have the lipoic acid. Remember that we talked about them as part of the gram-positive cell wall. But they could also be used for adhesion. And fimbriae, as we mentioned, and the pilus are many different organisms have them. Neisseria gonorrhea has pilae, Salmonella has fimbriae, and some E. coli also have some of those factors that allow it to bind to the epithelium. So they're going to have multiple factors allowing them to bind. Now, when they are bound to their tissue, they need to invade. So first, they come in, they attach themselves to the tissue. Now that they're there, they're going to start to dig into the tissue through invasion. And they need to penetrate the epithelium either in between the cells or inside the cell. Some cells of the immune system, in the M cell of the gut, is a cell that allows for transcytosis of material from the lumen of the gut inside the gut. And so microorganisms use that shuttling mechanism to go inside the epithelium of the gut. but most of the time they're going to try to invade by force. And remember, you have protein as well as carbohydrate structures. So what way do they do it? They break that down. So that's why they're going to make enzymes like collagenase, elastase, to break those protein barriers of collagen and elastase that are present in the connective tissue to prevent them from going in. They can, part of the connective tissue is going to have hyaluronic acid, and a hyaluronidase, excuse me, it's going to break down the hyaluronic acid, allowing microorganisms to now have a way to go inside and move through connective tissue. And lecithinase, those of you who took the lab, remember that lecithinase is an enzyme that breaks down lecithin, which is a phospholipid, and that allows the phosphatidylcholine to be broken down in the membrane so that microorganisms can invade inside a cell. So as you see, a lot of the exoenzymes that could be used for degradation of food could also be used for invasiveness. Yes, Anne?
How does the clot formation around the point of entry detect the Staphylococcus aureus from the host events? I'm trying to picture that mechanism.
So, in particular in the skin, when you have a the little cascarita, the little
Scab.
thank you, cannot think of it in English. The little scab forms, and that is part of a clot. So using a coagulase that is able to break some of that tissue allows the microorganism to get inside of it.
So coagulase breaks the clot.
Yeah.
It doesn't actually break, because the wording just says clot formation, so it makes it sound like coagulate just makes the clot.
No, it doesn't melt. It does, as far as I know, it doesn't help make the clot. It helps to break that down. Yeah. So now, once it invades, now they need to colonize and grow. And here, it's the interesting part because the initial bacteria that infect us are usually not sufficient to cause disease. You may have a really low burden of microorganisms in a glass of water that you had, and that low burden of microorganisms, for example, with Vibrio cholera, is not going to give you cholera. It is when the organisms have actually attached, invade, and now begin to grow that they can now get sufficient numbers to control infection. And in the case of a lot of microorganisms, they're going to use the quorum sensing mechanisms that we learned about in order to now activate the pathogenic genes altogether when they have reached sufficient numbers. We discussed the fact that Vibrio cholerae doesn't turn on the production of its own toxin until they have reached sufficient numbers to activate quorum sensing. So all the microorganisms are going to eventually simultaneously turn on the production of toxin, which is going to be secreted, and that is going to help now the organism escape the intestinal tract and move outside to taint another source of water. So the system of the toxin is helping actually move the organism from one host to the next, but only when a sufficiently large infection has been established. Now, as you know, nutrients are going to be limiting. Think about iron. In our bodies, iron is not floating around as a free molecule. It's always bound as a coenzyme to another protein. Heme, for example. Lactoferrin, transferrin, all those proteins have iron bound to it. So a microorganism that needs iron will use their siderophores, remember we discussed siderophores earlier, to be able to remove the iron from heme or transferrin or lactoferrin to now use it in its own proteins. So those things that we discussed earlier in class, they're all going to come back as material that you need to review. So here with the issue about the lactoferrin and transferrin used with iron availability. Now, as I mentioned to you, in the case of syphilis starts, for example, as a localized infection in the urogenital tract of the person who got infected, and eventually it goes systemic. Once it gets systemic, it will invade other tissues, in particular the brain. So having an initial canker in the penis, for example, of a person who got syphilis, allows sufficient bacteria to let them then invade to go into a secondary tissue later. So that will cause then the secondary systemic infection. And just as a terminology, whenever we have now bacteria in our blood, we have entered a state of bacteremia. Bacteria in our blood. And bacteremia is a normal condition that we always have. Every time you go to the dentist and they do dental work on you, you get a degree of bacteremia. I am a bleeder when I go to the dentist. So every time that they're working on me and I bleed, the microorganisms in my mouth, they get into my circulatory system. That is why oftentimes with people, have you heard about people who are going to have cardiac process that they tell them not to have their dental work? So yes, part of the issue is that a lot of bacteria could then form the immune system with the antibodies and clotting proteins are going to trap those bacteria into complexes, networks. Those networks are floating through our circulatory system, and guess where they land? In the heart. They land in the heart and they land in the valves of the heart. As you know, the valve opens up and closes down. Now you have a complex of bacteria plus antibodies that are going to activate complement, which is that ancient system that I was mentioning to you. Now you can make a hole on the valve system. So oftentimes people who are going to get heart surgery, they give them severe antibiotics before the mouth or have them do that so they do not get problems secondary to the dental work that they have done. So now, let's look at virulence as a whole. Because now you know that the microorganisms you have to be exposed to them. After you're exposed to them, the microorganism needs to adhere, they need to invade, and they need to then colonize. Exposure, adherence, invasion, colonization. So those are the 4 steps that we're going to go over and over and over again for the rest of the class. The entire class until we finish the class. So now, those mechanisms, those 4 steps are controlled by virulence factors. And as I mentioned to you, the ability of a pathogen to cause disease is related to their virulence. And we tend to think about it as numbers. Very virulent microorganisms, you can be exposed with 10 of them, and that will be sufficient to kill an individual. Versus other ones, you have to have millions of them be exposed to them in order for them to kill an organism. So highly virulent organisms are going to cause really severe disease, whereas virulent organisms cause little disease. And this is one of the advantages that we take during vaccination. We can take a virulent microorganism. And like in the case of the polio vaccine, polio is very virulent as a virus. We can attenuate that, which is basically removing their virulence and render that avirulent and use that organism now as a vaccination. So the attenuation process lowers the level of virulence of an organism. And virulence is a process that evolves because one of the things that we're going to learn is that the more virulent an organism is, the faster it can kill the host. And that is detrimental to the organism. Think of Ebola. Ebola infecting people, kill them immediately. That prevents infection from happening to new organisms. So you have to establish a level of virulence that is going to allow the organism to be spread to the next host. A virulent organism cannot really spread very well, and therefore you do not have that problem. So you always have to think about the evolution of virulence as the tendency of if new hosts are available, you get high virulence. And then when fewer hosts are available, the virulence is going to go down. And I'm going to eventually show you the case of the Australian myxovirus experiment, in which the Australian government decided to use a virus as a way of biologically controlling the epidemic, quote-unquote, of rabbits that they had. How many of you know about the rabbits in Australia? Some of you do. So we're going to look at the fact that when the virus was released in the population, it killed 90-something% of the rabbits, and it was super virulent. But then, when there were no more rabbits to kill, the virulence of the virus went down, which allowed now the rabbits to come back up. So biological control has to be carefully monitored because it can backfire. So again, the virulence of a pathogen tends to increase when the new hosts are readily available, like in the case of the rabbits, many rabbits, high virulence virus. It goes down when the hosts are limited, which is after the virus killed most of the rabbits. And this goes back to the point that I was mentioned to you of attenuation, the decrease of virulence of a pathogen. And we do it in the lab by passaging the virus in the experimental conditions to change the capacity of it to adhere. For example, poliovirus was passaged, it was made to infect monkey kidney cells in tissue culture, and the poliovirus is not really a virus that is assigned to infect monkey kidney cells. And with time, it became really good at infecting the monkey kidney cells, but that made it less virulent for us. And that was the virus that was used as the oral vaccine against polio. So oftentimes you can do it permanently because you can create a strain that is now permanently attenuated and it will not revert back, but that doesn't always happen. So that was a good example in the mumps and the measles vaccines, but with polio virus had some reversions which caused the polio vaccine associated poliomyelitis. And that was like 1 in 700,000 individuals who will get polio disease from the attenuated vaccine because the vaccine reverted and became virulent. So there was that very small chance, and the chance was calculated, and the vaccine saved a lot of lives, but 1 in about 700,000 people developed polio from the vaccine itself. Yes?
How do you prevent reversion?
Oftentimes, to prevent reversion, you have to engineer the vaccine in that regard. Attenuation accumulates genetic changes that are going to make it less virulent, but because you're naturally accumulating changes, you can revert that. Yeah, and that's the case of the polio vaccine, I think. So now, let's look at this issue with virulence. And here's 2 examples of bacteria. One, Streptococcus pneumoniae, and the other one is going to be Salmonella typhimurium. So what we determine here is what is called the LD50. LD for lethal dose. So a lethal dose of organisms that will kill 50% of the populations that you have. And the LD50, when you, for example, look at Streptococcus pneumoniae, which is, remember, we talked about that in class as the bacteria that was used to determine that transformation happened. So here you have a logarithmic scale. So you can inject with 10 to the 1, and that is how many organisms? 10. There you go. Or you can go over here with 50. When you get to 100 bacteria per animal, you have basically killed almost 100% of the animals. So 100 bacteria injected into an animal will kill it. So the LD50 here comes to a dose right there of about 50. That looks like 50? Yeah, maybe 50. They didn't put the scale, so I don't know exactly what it is. Compare that now with Salmonella typhimurium. Again, it causes typhoid. How many of you went to the meeting with Professor Erkin? So, really good talk. So you know the disease that she's talking about. To get a lethal dose with Salmonella typhimurium, you have to infect with 1 million bacteria. 100 bacteria of Streptococcus pneumoniae can kill a mouse. To get the same amount of lethality, you have to inject 1 million Salmonella typhimurium. So the LD50 of it goes somewhere around there to, I'm going to say, 5,000 bacteria required. So from this kind of graph, you can conclude that Streptococcus pneumoniae is a lot more virulent than Salmonella typhimurium. So when you think about this, you have 2 issues. Number one, we're going back to the point of toxicity. How How good is that bacteria to cause toxic events, like for example in the case of having an actual toxin, or how good is that microorganism at invading the tissue that is going to be required to cause damage? So those are the 2 points to be looking at. Now, let's look at toxins for a moment and some of these virulence factors. And as I mentioned to you, a lot of these virulence factors are going to be encoded by a plasmid. We all have E. coli, but we are not all of them suffering from enterocolitis from E. coli. Because that E. coli that we have doesn't have a pathogenic plasmid on them. But E. coli that are pathogenic, they have a plasmid that encodes the enterotoxin. We're going to look at it as an AB type of toxin, and that toxin is going to cause the diarrhea that can eventually kill a person by dehydration and shock. Clostridium tetani are going to have tetanotoxin, which is a neurotoxin, causing the disease tetanus, again in a plasmid. So Staphylococcus, especially Staph aureus, they get through a plasmic antibiotic resistance. And Staph aureus can cause a bunch of different diseases depending on what broader infection it is. But what we're going to bring out, is that a lot of these virulence factors are not innate to the organisms per se, but they have inherited them from either a plasmid or from a phage. We talk about lysogenic conversion. So that is a phage that when it infects a certain bacteria is given a gene, and those genes add to the virulence reservoir of the pathogen. So let's take a quick look at some of the toxins now. And we're going to divide these two into the exotoxins and the endotoxin. Exotoxins are secreted toxins by the microorganisms. Endotoxins are usually the LPS that is shed from microorganisms of the gram-negative family. And when we look at them, remember the chemical properties of them. So you have a table over here that goes by property and both the exo and endotoxins. These guys, endotoxins, they're LPS. So they are the lipopolysaccharide, highly pyrogenic, that is going to cause fever, but not very immunogenic. Only certain cells are able to respond to an immune response. This one's being proteins. They are heat labile, therefore they can be destroyed by heat, and they are very immunogenic when they're released. So now, what do we do? So depending on the mode of action, the endotoxin is the same. It's usually fever, diarrhea, and vomiting. But the other toxin is going to be depending on what mechanism that protein is targeting. In tetanus, it's going to be a neurotoxin that's going to cause the contraction of the muscle. In botulism, it's going to be the opposite. It's going to induce the relaxation of the muscle. So you have the flaccid paralysis. The toxicity of them, the exotoxins are super toxic compared to the endotoxins, which are minorly toxic. Because it is a protein, the exotoxins are really good immunogens. So we can actually use them attenuated or modified as vaccination, versus the endotoxins are not. And toxic potential, basically, can you modify that to use immunologically? Yes, with the regular exotoxin, but definitely not with the endotoxin. And fever potential, we call that pyrogenesis. Is it pyrogenic? The endotoxin is usually very pyrogenic. Fever is one of the most problems, and the exotoxins are usually not. And now when you think about where are they coming from, the exotoxins are usually encoded on an extra chromosomal element, either the phage or the plasmid, versus the LPS is encoded in the chromosome. So it is a chromosome-based gene. So now LPS is really funny because, as I mentioned to you, it's generated by a lot of gram-negative microorganisms. And here what I have for you is the horseshoe crab. The horseshoe crab has blue blood. And what we do with these poor guys is that we bleed them to almost death to isolate the amoebocytes that you can see over here. And the amoebocytes are very susceptible to LPS. So what you can do is a test to test for the capacity of a particular substance to kill the amoebocytes. And that's when you determine the amount of LPS present in a solution. So you can either measure that by seeing the coagulation being generated, or you can look at it by measure spectrophotometrically to how many amoebocytes you have present. And we call this the limulus test. These guys are limulus. That's the species of the horseshoe crab. And for these guys, they're not going back into the ocean. Nowadays, I think we're releasing them back. We're not removing their tails like they did in this case, but as you can see, they took out the abdomen of them. They keep the thorax and the head present, and they put a tab in their bleed, and they bleed them down. Yes?
Very off topic, but why is their blood blue?
It's a different chromogen in their hemoglobin that makes it to be blue instead of red.
So like when it's oxygenated, it will turn blue as opposed to red.
It's always blue. It's different shades of blue. So these are the only guys that I know that are actually blue bloods. The aristocrats, it doesn't have blue blood as we used to call them. Yes, Anne?
You said that they bleed them almost to death, so what do you mean almost to death?
Depending on the mechanism and who's doing this now, there are companies which are bleeding them like they're doing blood donations. So they trap them, they tap them, they get blood, they release them back, let them recover, and then they harvest them one more time. So the little horseshoe crabs, which are not crabs per se, they're actually arachnids. They're of the spider family instead of the crustacean family. They're closer to the spiders. They have that, and we use their limulus. So now, I want to show you that. Now, let's talk about the exotoxin because they're actually fascinating. And here what we have are the proteins being released. And mostly, either you're going to be cytolytic toxins, which are able to destroy something. Those of you taking lab, we talk about the streptolysing O, that are able to destroy red blood cells directly, and that is the one that we use in the blood agar to look at the clearings of hemolysis around the blood. But you also have a different type of toxin architecture, which is the AB toxin. It's a two-component toxin, A for the outside active part and B for the binding part. Again, architecturally speaking, we can look at that later, but most likely the active part of the toxin gets injected into the cell after the binding part of the toxin binds to the outside of the cell. And last but not least, you have the superantigens. The superantigens are called such because they are proteins that can cross-link the T cell receptor, and when they cross-link the T cell receptor without another molecule to give this receptor a signal, they can cause the death of the T cell. So we use those in immunological research because they can cause a great amount of inflammation by overstimulating the immune system. Now, this table has all the kinds of toxins that you may possibly want. So here's diphtheria, botulism, perfringens, etc., etc. I want you to take a look at what they are doing and which of the pathways are being affected, again, as a way for you to get used to it. We're going to take a look at some of these really important toxins like Bordella pertussis, which is the whooping cough toxin. It is a G protein blocker. Remember G proteins from Bio 110. And the other one that I want to bring up to you is the Diptheria Toxin, which is an AB type of toxin, and that is going to inhibit protein synthesis. In this table, if it's a cytotoxin, you have CT. If it's an AB type of toxin, you have AB. And if it is a lymphocyte toxin, you have LF over there. And the last, here you have a bunch of different, this is Staph aureus. This is the flesh-eating bacteria, which is now resistant to all kinds of antibiotics. Notice the number of toxic factors that it has. So it has a huge arsenal that can eventually be very problematic depending on what it's doing. So now, diphtheria toxin, it's expressed by the bacteria Corynebacter diphtheriae. It's an AB type of toxin, and what it does is that it prevents translation. So remember that during translation, you're going to have the EF2 factor that is going to bring the tRNAs down to the ribosome to allow the peptide bond to be made from the new amino acid to the chain that is already growing inside the ribosome. But the AB toxin, what it's going to do is to sequester away the EF2 factor and therefore now preventing the tRNAs from coming into the ribosome. So it basically blocks translation. Now, the other two that I want to talk to you are the botulinum toxin and the tetanus toxin, because these are two AB toxins. We spent a lot of time in Bio 110 learning about how contraction of a muscle happens. And we talked about the fact that when the neuron releases acetylcholine, the acetylcholine will bind to the acetylcholine receptor, and that is going to induce a depolarization of the membrane that is going to induce an action potential. So here you have in this example on the left. So here you have your neuron releasing acetylcholine, and acetylcholine can now bind to the acetylcholine receptor and induce constriction of the muscle. The botulinum toxin, Botox, that now we use cosmetically, prevents the release of acetylcholine. And because it prevents release from acetylcholine, it is basically blocking the contraction of the muscle. That's why we're using cosmetically, or in other instances, actually medically, to prevent effects that are muscle related. So it induces flaccid paralysis, excuse me. On the other hand, before I go there, excuse me, the problem is, remember, as we breathe, the chest muscles in the ribs are helping us contract the chest cavity to allow us to breathe. So if now that muscle cannot contract, we cannot breathe anymore. That is part of the botulism issue. Now, tetanus, on the other hand, is interesting because part of the issue that we had discussed in Bio 110, by an inhibitory neuron, the inhibitory neuron can secrete, for example, glycine, and the glycine is going to bind to the glycine receptors, allowing chlorine to come inside, and that changes the polarity of the neuron, which is going to prevent it from releasing acetylcholine. Now what you have with tetanus, it's the prevention of the release of glycine by the inhibitory neuron. So glycine cannot be released, therefore the neuron is constantly releasing acetylcholine to the target muscle cell, and the muscle cell goes into a constant, permanent constriction. And this is what we call the tetanus state. So this individual here that you can see is undergoing tetanus. So every single muscle of his body is contracted, bicep contracted, chest contracted, the back muscles contract and the neck muscles contract, so the person arches up. And that prevents the normal function of the body, and it could be quite lethal, especially. Now, the last toxin that I'll talk about in the next couple of minutes is going to be the cholera toxin. And the cholera toxin is released from Vibrio cholerae, which is a gram-negative microorganism shown over here. Now, this toxin is super interesting because it's going to affect the adenyl cyclase. As you know, adenyl cyclase mediates the capacity of a cell to make cyclic AMP. So here, during normal conditions, you have the lumen of the intestine, and here's your intestinal cell, and there you go with your blood, and sodium, for example, can come in through it. This toxin is an AB type of toxin. So here you have the two portions of it, the AB, which is the active portion, the B is the binding, and it will bind to the receptor, the GM1 receptor, and go inside now the A portion of it. The B portion stays outside. Now that is going to affect adenyl cyclase, which is going to create a great amount of cyclic AMP. That great amount of cyclic AMP is going to prevent the movement of sodium inside, and is going to get carbonate and chlorine to go out. This imbalance of electrolytes, which is basically caused because, remember, cyclic AMP is activating phosphokinase A. That is going to now cause the diarrhea, and that diarrhea is what the microorganism is going to use to access the bowel of the host. So take a look in this, and I will finish the lecture with this graph, this little image, which is showing you the bacteria with all its arsenal of goodies. The plasmid that has virulence, the pathogenic islands in its own chromosome, the O antigens, for example, the flagellum, the cilia, the villi, all those things are going to allow it to move. What I included in this lecture for your amusement are a set of slides that are going to be at the end, and they are going to have a lot more interesting and detailed parts about something. I used to divide this into two classes and have them all. So you can take a look at these slides and learn from them as well. Some of them are going to be about the skin, Other ones are going to be about the oral cavity, in particular the generation of plaque around the teeth, I know, and the bacteria that are involved into doing that, as well as the bacteria that are involved in the gut and the gut pathogens. So I will leave you with that, and I will then see you on Tuesday of next week. Have an excellent Thanksgiving, and have fun, guys.